Hello everyone, my name is Eric Yanikomshan. I'm a CBN major at UC San Diego, and today I'm going to be talking about CNTNAP2, a gene that is involved with autism spectrum disorder. So, quick background. A couple weeks back, I was at the Living Room, which is a cafe in downtown La Jolla, and I overheard a couple of people talking about hippocampal neuronal connectivity and just topics in neuroscience that piqued my interest. As a neuroscience major myself, I decided to go talk to them. And long story short, these gentlemen worked in a company that helps children with autism. And they told me about this gene called CNTNAP2. Now these guys, they're audio engineers, and they're working to make a treatment in that way. So I decided that with the UCSD resources that I have, let me see what I can bring to the table. So, ASD. It's a neurodevelopmental disorder that characterizes deficits in language development, social interaction, and prolonged repetitive behaviors. Modern literature does not classify ASD as a disease, but rather as a developmental disconnection syndrome due to abnormal deficits in cortical connectivity. So what does CNTNAP2 actually do? Well, it's a gene which codes for a protein called Caspr2. And according to an article by researcher Olga Peña-Garakino, Caspr2 plays a prominent role in cell-to-cell signaling, nervous system development, organizing and localizing potassium channels, and most importantly, it is highly expressed during embryonic and fetal development. CNTNAP2 mutations lead to neuronal migration difficulties, reduction of inhibitory neurons, and an imbalance of inhibitory and excitatory neurons, which lead to an overall abnormal neural architecture and abnormal neural synchrony. In situ hybridization experiments demonstrated that CNTNAP2 expression is found in regions of the brain such as the cerebral cortex, hippocampus, striatum, olfactory tract, and the cerebellar cortex. Now, these listed brain regions are responsible for a magnitude of cognitive and behavioral functions, making it clear that mutations lead to a pleiotropic effect, as in an effect that has a wide variety of phenotypes. Now, building off on this, a critical point that Olga emphasizes is that, quote, we must start viewing these genes as influencing development and function of brain circuits rather than having a direct effect on behavior. It is dysfunction of the circuits that may variably lead to clinical phenotypes, and this goes to show how the neuronal architecture plays a large role in one's cognitive and behavioral features. Switching to a different researcher, Sisay et al., hopefully I'm pronouncing that right, had CNTNAP2 knockout mice and found that the knockdown of the gene in layers 2 and 3 of pyramidal neurons in the prefrontal cortex decreased the number of functional synapses and elevated inhibition excitatory imbalances, as mentioned earlier. Such physiological differences led to behavioral phenotypes, the most notable of which showed a significant decrease in sniffing and social interaction. So, I know that was a lot of scientific jargon just thrown at you. Let's go ahead and understand that these social interaction tasks involve the mouse subject, which either has the gene or a knockdown. And these subjects are put in the same cage as another mouse who acts as a friend, aka the novel stimulus. And so the results showed that the mice without the CNTNAP2 gene, otherwise known as the knockouts, interacted significantly less with the friend than the mice with the gene itself, or the wild type. Now, these behavioral features are seen in the context of an experiment with mice models, but at the same time, many doctors, many researchers notice the same traits in humans as well. So what are the broader implications of these findings? We've discussed how abnormalities in this gene lead to an asocial phenotype per se, but how does the physiology affect the behavioral function? An important point to bring forward is the cortex itself. specifically the frontal cortex. Olga Peña-Garakino, in another study, compared the differences of CNTNAP2 knockout mice to wild types and utilized neuronal markers such as CUX1, FOXP2, and BRDU to identify the presence of cell types in the cortex. So briefly, CUX1 is a marker for neurons in the upper layers, 2 and 3. FOXP2 is a marker for neurons in the deeper layers, 5 and 6. And BRDU is a marker to identify newly formed ectopic neurons in a sense where they shouldn't be there. The findings of this analysis found an abnormal amount of CUX1 and BRDU markers in the deep cortical layers without the presence of any FOXP2. Now keep in mind, the wild types had the CUX1 in the higher layers and the FOXP2 in the deep layers. So there's a big difference. And so piecing together this puzzle, we see that these abnormal neural migratory patterns lead to an improper wiring of the brain itself. There's still a lot to unpack about CNTNAP2. From effects on GABAergic interneurons, to the imbalance between inhibitory and excitatory neurons, medications such as CX546 and risperidone, we have not been able to solve the whole puzzle. Further research will need to be done in order to start understanding the big picture behind autism spectrum disorder, and specifically the involvement of CNTNAP2. For now, a solid foundation has been placed, and I'm excited to dive deeper into this research and find some more solutions. Thank you so much for listening. I want to give a big shout out to Ryan from the living room in La Jolla for inspiring this project, and the next step is to look into the audio engineering stuff. Thank you so much, and stay tuned.